to welcome Professor Greg Lang from Lehigh University. He drove all the way up here from Bethlehem, Pennsylvania today. So I'm really appreciative. I actually met him this summer when I was a student in a class and he was the instructor. And what I came away with is that, number one, he's Mr. Genomics. Number two, he's not afraid of mathematics. Keep that in mind, you freshmen, in your calculus classes. And he's a great teacher, so I thought he'd be really great to come speak with us today. He was an undergraduate at Military University, and I have a feeling he did really well there. He majored in molecular biology, but he was also a minor in physics and biochemistry. And yeah, I'm pretty darn sure he did really well because then he got into Harvard University for graduate school. And when he was a graduate student in molecular biology, he studied mutation rates in yeast. And in fact, he perfected this technique to measure them that I'll be using in our genetics class next semester. And from there, he went to do a postdoc at Princeton University, where he began studying genomic evolution. And he's continued those studies, and that's what he's going to tell us about today in his talk, Genome Evolution in Laboratory Populations.
Thank you, Rachel. Thank you. It's nice to be here. I am a molecular biologist by training. I'm a card-carrying molecular biologist. My undergrad degree says, that's what my PhD says. But I've always been passionate about evolution. And so what my lab does is we use the tools of molecular biology and genomics to study the process of evolution in a technique that's known as experimental evolution, where we can watch this process in real time in laboratory populations. So what I hope to do today is to tell you about a lot of the things that we do in my lab and sort of impress upon you really the power of this method in understanding how genomes change, how genomes evolve. So I want to start though with an example from nature. So it's long been known that if you have parallel populations that are more or less identical but are evolving in the same environment, that they'll tend to find similar solutions to the same selective pressures. So a classic example of this are these cavefish populations. You can find these in Mexico and parts of the US Southwest. They exist in two different morphologies. So you have the one on the left that looks like your normal swimming freshwater fish. Periodically, these fish will find themselves trapped in a cave environment, and when they do, they'll adapt. And on the right, you see two populations that adapted independently to the same environment. And you can see they did so through similar morphological changes. So they've lost pigmentation, they've lost eyesight. And in fact, this parallelism extends even to the genetic level. So both of these populations have different mutations in the same gene, which underlies the loss of pigmentation. And so one of the driving questions for my work, really for the whole field of experimental evolution, is, how surprising is this? How reproducible do we expect evolution to be? If we can run exactly the same evolution experiment 100 times over from the same starting point, will we always get to the same ending point? Is evolution in any way predictable? And if it is, how far in the future could we forecast evolution? If we could forecast evolution, this would be revolutionary for understanding the evolution of antibiotic resistance. We can perhaps steer populations away from evolving antibiotic resistance. And so this question is really what drives and motivates the work in my lab. How reproducible do we expect evolution to be? Now, we don't study cavefish in my lab. My lab uses budding yeast, the same yeast you would use to bake bread or brew beer. It's a very common workhorse of molecular biology. This genome is very well understood, and we can manipulate it in very many ways. And the process we use, I already alluded to, is called experimental evolution. So what is experimental evolution? So you could imagine working in the lab and doing the following experiment. You can come into the lab one day, you can inoculate a test tube with some yeast or bacteria or whatever. You can let that grow to saturation. You can come back in the next day and dilute that culture back into fresh medium, let it grow to saturation, come back in the next day, dilute it again, and just do that over and over and over again. And that's essentially what we do in my lab. We don't actually grow our yeast in test tubes. We grow our yeast in 96 well plates, which are about this big. And this is, as it says, 96 wells. So we can have almost 100 populations in a very small footprint. And we actually don't even do the dilutions ourselves. In my lab, we have robotics. And so grad students will come in the morning. They'll set the deck up with the plates that had grown up overnight. They'll put empty plates on the deck. They'll push start, and the robot will dispense fresh media, resuspend all the cultures, and do all the transfers. Yeast grow very quickly, and so they get about 10 generations of growth per day. And so you can extrapolate and say that over the course of a year, we get well over 3,000 generations of growth. And so we can evolve hundreds or even thousands of populations for thousands or even tens of thousands of generations very rapidly. This tool, this technique of experimental evolution is very powerful. For instance, we can do these evolution experiments with many different starting points. We can evolve yeast that are haploid, meaning they have one copy of the genome. Much of what I'll tell you about today is doing just that. We can use haploids versus diploids. We can evolve other strains or other species of yeast. We can evolve hybrids and ask how quickly the hybrids adapt. We can change how we do the growth. So we could do experiments where we alternate the environment, where we pull them between two different carbon sources, for instance, every 100 generations. Or we could do an experiment where we just ramp our incubator up 1 degree every 100 generations and ask how quickly can evolution keep up with the changing environment. We can also do our evolution experiments with sexual populations and ask what is the effect of recombination on the rate of adaptation, on the reproducibility of evolution. We can change how we, or more appropriately, how the robot does the dilutions to change fundamental parameters such as the population size, or we could even introduce migration between adjacent wells and ask how does migration between neighboring isolated populations affect adaptation. And as we do these evolution experiments, we have a lot of tools at our disposal. One of the most important things is that we can maintain all of our populations frozen at -80. And so as we do an evolution experiment, we will periodically freeze down the entire 96-well plate, and we have our frozen fossil record. So we can go back to any population at any time point. We can pull it out of the freezer. We can look at it in the microscope. We can sequence its genome. We could even restart the evolution experiment from earlier times. One of the techniques that we use quite often is flow cytometry. And we use flow cytometry, as I'll explain in a slide or two, to ask how much more fit is an evolved strain. So after several thousand generations of evolution, how much better is this population from when it started? Well, we can go to our frozen fossil record, and we can directly compete head-to-head populations that are separated by thousands of generations. Probably the most powerful tool that we have in our arsenal is next-generation sequencing. Next-generation sequencing has become cheap enough and accessible enough that we can now take all of our populations and sequence the genomes of everything that's evolved and see exactly how our yeast have changed in response to whatever selective pressure they're under. So experimental evolution is a very powerful and very versatile tool that we can use for studying evolution in real time in the laboratory. And so what is some of the things we can look at when we do an evolution experiment? So I'm going to show you a few slides, and you're going to start seeing evolved populations. These are all haploid yeast, so one copy of the genome. They all evolve asexually in just rich glucose media, the standard thing that we use in a lab, at 30 degrees, which is the happy temperature for yeast. And so this is the simplest possible evolution experiment you can do. Here are 3 populations that evolved for 1,000 generations. And what you're looking at here is you're looking through the bottom of the 96-well plate. And so this is a settled pellet of yeast that are at the bottom of each of these wells. And this is, over the course of 1,000 generations, this is the same well in each of these cases. And I said you get 10 generations of growth per day. So between this time point and this time point, it was 14 transfers, which means the robot went in, mixed this culture, and transferred 1/1000th of that culture 14 times. And so sometimes we can see things that change just visibly by eye. And so the most obvious one is this middle column. You see this real kind of funky pellet morphology that seems to have swept through the population. So clearly something has evolved. And there are cases where you don't really see anything dramatic, but if you notice in the bottom one, it's pretty clear that the pellet size has gotten larger over the course of evolution. So they've gotten better, perhaps at consuming glucose that they're provided in this medium. But most of the time it actually looks kind of boring. So here, not much has really happened. Maybe the pellet's a little bit bigger. But as I'll show you in a little bit, we actually know that quite a bit is happening under the hood, even in populations that look like this. So that's the simplest thing we can do. Look at our cultures. There are slightly more complicated things we can do, and this is going to get back to this idea of a fitness assay and asking how much better is something quantitatively from where it started. So we can take our evolved population, we can mix it 50-50 with the fluorescently labeled version of its ancestor. So we have a version of the ancestor that is expressing GFP, and if we would put these two strains in a flow cytometer, you could distinguish the reference from the evolved. And if we start them off at 50-50, we can then propagate this 50-50 mixture over time, just as we would do during an evolution experiment. And in this case, we're directly competing these in co-culture. What I'm going to show you now is real data from one of those experiments. So on the x-axis is the generation of the competition. This is after we mixed these two strains 50-50. We competed them head-to-head for 50 generations. On the y-axis is the log ratio of the evolved strain to the fluorescent ancestor. And if you notice that since it's log of, if this is a 50-50 mixture, log of 1 is 0, so they start off close to 0. And this is real data. They all increase in frequency, meaning the evolved strain is outcompeting the ancestor. And we can measure this slope. In this case, the slope is 5.5 plus or minus 0.4%. So what this means is this evolved strain was 5.5% more fit than its ancestor. And we can measure that fitness to an accuracy of within about 0.5%. And so what a 5% fitness advantage means is if the ancestor is going to produce 100 daughter cells, the evolved will produce 105. And so it's a real subtle difference, but evolution can act on very subtle differences. So the most powerful thing that we can do, as I mentioned, is sequence these genomes to figure out what's going on. And so just to come back to this slide, we can use these fancy instruments. This is an Illumina sequencer. And we can sequence, for instance, this population. So these were, again, as I said, haploid yeast. There's only one copy of the genome. It was completely asexual. And we sequenced some of these genomes. And what we did is we sequenced the entire population. So we took everything that's in the well. We sequenced the entire genome, the entire population, and we did this over the entire time course of 1,000 generations. So what I'm going to show you in the next slide is what that data would look like. So here's a single population. And what you see with each of these traces is every mutation that we could detect within that population. And you'll notice some mutations come up and drop out. That's because this is an asexual population. So if you have two beneficial mutations that are moving through the population at once, because they can't recombine, only one of them can ultimately win. And so a lot of beneficial mutations will go extinct. We can show this data in a slightly different way. These are called Muller plots. What you see here is the exact same data, but now shown in such a way where the width, the height of these different colors reflect the abundance of these mutations in the population. So here, you had two mutations in GAS1 and LAP4 that spread through the population and fixed, so then every individual had them. And then within that background, you started getting additional mutations. So here's another such population like that. This is a little more complicated, where you have more mutations. The majority genotype at generation 500 ultimately went extinct. But this gives you an idea of the type of data that we can get by applying next generation sequencing to our populations. We can see all the mutations that have arisen over the course of 1,000 generations. We've done this extensively, and so one of the major projects we did is we sequenced the entire population for 40 of our evolved populations. And that data is shown here. I don't expect you to see much from this, but I want to make a few points. One is that every one of these populations looks different. So at the molecular level, at the level of what genes and what mutations you see come up, they're all unique. And I like to think of these as 40 case studies of evolution. And so from these 40 case studies, we can begin to learn great detail about how evolution happens. And remember, this is just a really simple, well-controlled population. Just a single well of a 96-well plate that we've been transferring every single day. What I want to do in the next few slides is walk you through a couple examples of the kind of things we can learn from this type of data. So one of the first things we noticed when we started doing extensive sequencing of our populations is that when mutations come up, they rarely come up as individual mutations. And you may have seen this on the slide that I showed previously. You know, here we have a group of 5 mutations, and we've called these cohorts, really for lack of a better term. It was actually kind of amusing how we try to come up with the term for these groups of mutations moving through the populations, and this term we found out has now even made its way into evolution textbooks. So it's kind of strange when I go to conferences and hear people using this term of cohorts of mutations. But what you see is a cohort of 5 mutations that arises and sweeps through the population. Several hundred generations later, you have this cohort of 3 mutations, followed very quickly by this cohort of 5 mutations. Another thing that we noticed when we started looking at sequencing a lot of these populations is that we tend to see the same genes mutated over and over again in different populations. So in this population, there's a single mutation, which is in PDR5, which is spreading through the population. It gets alpha-beta by this cohort of 6 mutations, which you can see in orange. That in turn gets alpha-beta by this cohort of 9 mutations. Now, if you look carefully enough, there's a mutation in KRA6 in both this orange and this yellow cohort. These are 2 different alleles, 2 different mutations within the same gene. And if you look at generation 600, you would see one KRA6 mutant allele is spreading through the population, and one is going extinct. We also saw fairly complicated dynamics within these populations. And so we saw a number of populations where we got mutations that rose to a certain frequency and then just stayed there, stably for hundreds of generations. And in these populations, we can even see selective sweeps happening within these subpopulations. So again, in a real simple environment, just a single well of a 96-well plate, we actually get some very basic sort of ecology arising where we get the population splits into subpopulations. And so this is really nice. It gives you a good idea of sort of the dynamics of adaptation. They're very pretty pictures. You can see lots of mutations coming up, lots of getting repeated. You can say something about how adaptation works in asexual populations. But what we really wanted to know is, what are the mutations that are driving adaptation? What is really being selected for in these populations? And to get at that, I want to come back to the point that I made in the middle, that we tend to see the same genes hit by mutation over and over again across these populations. And so, just to do a little simple statistics, the yeast genome has about 5,800 genes. Across all of the sequencing we've done, we identified 723 mutations that hit a gene. Now, if you just take 723 mutations and throw them randomly across 5,800 genes, you would expect that most of those darts or whatever would hit a unique gene. You would expect to hit 40 genes twice by chance, but you would expect to hit only 2 genes 3 or more times by chance. So that's the null expectation for throwing darts across the board, throwing mutations across the genome. What we see, however, is a very skewed distribution, where there are 24 genes that account for a vast majority of the mutations we observe. So just to illustrate that, these 24 genes that we see hit more than 3 times by mutation, they represent only 0.6% of the yeast genome by size, yet they account for 14% of the mutations we observe. So these genes are likely the genes that evolution is selecting for mutations to arise. So what are these 24 genes? So this is a list of the genes that we see mutated more often than we would expect by chance in our simple evolution experiment, which is rich glucose food. And so these are the biological processes that are likely driving adaptation. Leading the list is IRA1. We see it mutated 21 times across the 40 populations we selected, so about half the populations get an IRA1 mutation. We can also look at the biological processes that we see mutated. We see things like the mating pathway, and we know why we see that, because if it's a haploid yeast growing asexually, there's actually a cost to being competent to mate. Yeast are pumping out lots of mating pheromone to attract a mating partner. If one is never going to arrive, it's actually beneficial to just shut off that pathway and save that unnecessary protein production. We also see a lot of mutations in the RAS pathway. And so what we think is happening here is the RAS pathway is responsible for slowing down cell growth as you're running out of sugar. And in the lab, as long as we faithfully dilute them back every 24 hours, it makes sense for them to take their foot off the brake and just start growing as fast as they can. And we actually know that if we then starve them, allow them to go into stationary phase, these IRA1 mutants are actually quite sick and dead. And we also see a lot of mutations in cell wall assembly and cell wall biogenesis. And later in the talk, I'll get to an explanation as to why we think those mutations are arising. Just to show that these are really the drivers of adaptation, for the 141 mutations that hit these 24 genes, only one of them is synonymous, compared to 18% synonymous mutations for everything else we see. So when we are mutating these genes, we are changing their function. Because we sequence them all, we actually know exactly how we're changing their function. So these are the 121 IRA1 mutations. You see a mix of nonsense, so premature stop codon, frame shift mutation, missense mutations, and so we see all sorts of mutations. So essentially, when we are mutating IRA1, we are killing its function. We are selecting for loss of function of these negative regulators of RAS. In contrast, these cell wall assembly proteins, we only see missense mutations. So this suggests that for these genes, we're selecting for either alteration or attenuation, but not loss of function. And if we go into our yeast and we delete GAS1, the cells are sick. If we delete KRE6, they're dead. And so we know that these are alteration of function mutations. And so this tells us now which genes, what are the genetic targets of selection in our evolution experiments? But what it doesn't tell us is how much of a benefit do they get for shutting off the RAS pathway. So what is the actual fitness effect of these different mutations? And to get at that, we can turn to what is another real powerful aspect of our system. The fact that yeast is a great model system for doing genetics. And so we can do genetic crosses with our evolved populations. And I had a postdoc in the lab who developed a very clever process for doing really high throughput genetics in these populations. So here's a cartoon example of what an evolutionary trajectory might look like. And the ancestor, over time, would pick up 5 mutations. And let's say that this evolved population has a 5% fitness advantage. What we want to know isn't that all 5 of these mutations give you a 1% benefit, and they all just add together to give you a 5% effect fitness advantage. Is it that there's one 5% mutation, and then everything else is neutral? Or is it something more complicated, where you need the red, the yellow, and the blue to get any benefit, in which case they add together and sort of they combine in a non-additive way? And so to understand which of these are actually the drivers of adaptation and to quantify how they affect fitness, we can take our evolved population and we can cross it to an ancestor of the opposite mating type. If we do this, we now make a diploid strain that's heterozygous for all of the evolved mutations. And we set this cross up such that this diploid strain is also heterozygous for the dominant drug sensitivity gene CAN1 at 2 unlinked loci. And so we can kill these diploids by adding the drug cannabinine. And if we sporulate this diploid, 3/4 of those spores will also have a functional CAN1 and will also be dead. But this provides an efficient way of selecting for haploids at the other. We did some tricks that we can do in the yeast genetics field, where we convert this diploid to MAT-AA, meaning that it only has one copy of the mating type locus, and only one type of haploid progeny can come out. We can sporulate this, and we can plate this on media that contains the drug cannabinine. So we have these really large petri dishes that contain the drug cannabinine. We can take these diploids that, remember, are heterozygous for all of the evolved mutations. We can plate them as a lawn, and we can get hundreds of thousands of colonies that grow up. All of those are going to be identical to the evolved, but they will have random combinations of the evolved mutations. And so I like to say that we're essentially using Mendel to shuffle the deck and make all possible random combinations of the evolved mutations that were present in this clone. Once we do this, we can get these very large pools of segregants. We can then put them in one tube, or in our case, one well of a 96 well plate. And we can propagate that well forward over time, just like we would do during an evolution experiment or during a competition experiment. And when we set this up, initially, every evolved mutation will be present at 50%, and all of these genotypes will be random. When we expose it to selection by propagating it forward, any beneficial mutation will enrich in frequency in the pool. Any neutral mutation will stay at the same frequency at 50%, and if it's a deleterious mutation, it will actually drop out. And so this is a very quantitative and a very high throughput way of measuring the fitness effect of all of the mutations that are present in any one of our evolved clones. And so here's a very simple population where we do this. So here's an evolved population where we had two mutations that fixed early on, ELO1 and GAS1. This is a cohort of two mutations, followed by a mutation in sterol 12. Taking this strain, we isolated a clone from generation 400 to 500. We back-crossed it. We made a large pool of segregants that contained all random combinations of just these three mutations. And then we propagated that pool forward over time. And so this is now the competition of propagating that pool forward. What I want to point out is this ELO1 mutation did not enrich in the pool, meaning that it was a neutral mutation. However, both the sterol 12 mutation and the GAS1 increased in this pool when we essentially used Mendel to shuffle the deck. And so we can quantify the fitness effect conferred by these two mutations. And then we infer that the ELO1 mutation only fixed in the population because it was associated with a known driver mutation. So this is what we would call a hitchhiker. So this is a simple population. We can do this for populations that are a little more complex. So here's a population where by generation 1000, I think we had 24 mutations fixed within this population. So we took a clone from generation 1000, we back-crossed it to the ancestor, we got a large pool of segregants, and we propagated that pool forward over time. And this is what that data looks like. What I want to show you here is that most of these lines are flat. What that means is that most of the mutations that fixed in this population do not confer a selective benefit. If I highlight only those mutations that actually have a significant slope, you can see that there are 3 of them, ROT2, IRA1, and MID2. And we can map these back onto the dynamics of adaptation, and we can see that each of these driver mutations correspond to one of the main selective sweeps within this population. So this is consistent with the selective sweep being a single beneficial driver mutation and anywhere from 1 to 10 hitchhiker mutations. We did the same construction of these pools in quantifying the fitness effect of all of the mutations in our evolved background for 11 different of our evolved populations. What I'm showing you here is the 11 populations identified at the top and separated by these dashed lines. And then I've highlighted, this is the fitness effect of all of the mutations within a population. This is that one where it's ELO1, GAS1, and sterol 12. And in red, I've highlighted all of the mutations whose slope differs from 0. And so you can see that most of the mutations that fix in our evolved populations are neutral. One of them appears to be deleterious, which is kind of interesting. And we can identify, fortunately we at least identified one beneficial mutation per population. we can say something about what these beneficial mutations are. Before I made a statistical argument, I said we can guess which genes are likely beneficial.